Hello, and welcome to Arcadia University's BI327 Histology course. In this first lecture, we're going to take a look at Introduction to Microscopy. And in the first portion of this lecture, we're going to look at some basic properties associated with microscopy, namely that of magnification and resolution. As you can see, with all of the different topics within the course, we're going to have a list of objectives that help identify what are the goals associated with the topic, as well as provide an opportunity for you as a student to understand what I, as the instructor, is seeing as the most important components associated with this. And again, these will be available on the web page, they're in the syllabus, and they're going to be available on the course website as well. Now, when we take a look at this concept of histology, or essentially what is the topic of this course, microscopic anatomy, what we're going to be looking at in essence is going to be the relationship between structure and function within the body. And so if we think about this in terms that we've already kind of talked about in other courses, structure is in essence anatomy. It's how things are built. Function is going to be the physiology component. It's basically how things are going to be working. And hopefully as you go through this course, what you're going to understand is that there's a very strong relationship between structure and function. Things look the way they do because they do certain things. Things work functionally because they have the correct structure associated with it. So if we were to step outside of the human body, step out of a biological system, and take a look at other examples of structure and function, one of the prominent examples is to take a look at buildings in the area. If you look at the image on the left-hand side of the screen, you see a warehouse. You see a building that has a lot of floor space, but in essence really only has one story. Maybe have a second story for maybe some offices in some location, but everything is accessible on that first floor. You compare that with the skyscraper on the right-hand side of this image, right-hand side of this slide. And what you can see is still a lot of floor space, a lot of space available for use, but it doesn't take up a lot of space on the ground. And so by looking at this, the warehouse on the left or the skyscraper on the right, we can make some assumptions about what's going on both within these structures, within the buildings that we're looking at, as well as what's going on around them. If we look at the warehouse, we can say, okay, this is probably an area where land values are relatively cheap because we're looking at a building that spreads out across a long portion of the land. In contrast, in the Center City, Philadelphia region, the costs of land are going to be very high. So what you're going to have is a relatively small footprint for this building, and it's going to grow up as opposed to out across the land. If we take a look at the skyscraper, we're essentially going to see it's like floor upon floor upon floor. And so internal to this building, we can expect to find some type of mechanism for getting people from the entry level, the ground floor level, up to what we would expect to find offices on the upper levels. And so we can expect to find, a staircase or multiple staircases throughout this building. We're going to expect to find an elevator as a system involved with rapidly moving individuals back and forth within this structure. In contrast, if we look at the warehouse on the left-hand side, maybe it has a staircase if we got some offices on a second floor that we don't obviously see from this view of it, but we probably don't have an elevator going on there. So again, you can see the relationship between structure and function when we take a look at these two images. When we take a look at moving back into the human body, what we're going to see is again that relationship between structure and function. And it's going to be important that we recognize that hierarchical relationship of what's occurring within the body. In the basic anatomy courses that you've probably taken up to this point, you've talked about organ systems. So you're talking about the digestive system, you're talking about the urinary system, and maybe get a little bit of detail about the specific organs that are found within it. So within the digestive system, you've got the esophagus for delivering food to the stomach for starting that process of digestion, passing the materials into the small intestine. which is involved with additional digestion and absorption of the materials. What we're going to do within this course is still look at these as organ systems, look at these as organs, but focus in more on what's going on at the level of the tissues, the cells, and the molecules. So what's going on to actually conduct these basic processes of life to adapt, to allow an individual to survive, and to essentially provide the functions that are necessary, the physiological functions, for the continuation of life. So if we take a look at this, again, some terms that we can describe this course. Anatomy, as we talked about earlier, is the study of body structures. Physiology, the study of the functioning. What we've got is essentially in this histology course is something that's going to merge together the gross anatomy, what we can describe by looking at it with the visible eye, the kind of larger structure, and combine that in terms with microscopic anatomy, using a microscope or using some type of mechanism that we can study what's going on in a much finer detail within the body. And in other courses, this would be molecular biology, the study of molecules, cytology, the study of cells, or this course is called histology, the study of tissues, and the way we're going to approach it is, in essence, to look at all of these different things. So in order to be able to study this at the microscopic level, we have to use this concept of magnification. We have to increase the apparent size of the specimen, of the image that it is we're looking at. Now, it's important to keep in mind that basically what we're using is a series of tools that are going to allow us to see something at greater and greater magnification. So we're making the image bigger, we're not making the object bigger. Now, magnification in and of itself is valuable, but it's important that we magnify the image and we don't make it fuzzy, because we want to have a nice, clear image that's visible, as opposed to a really large fuzzy image that we can't determine what's present within it. So when we talk about magnification factor, and at least at this portion of the course, we're not doing essentially microscope work, but it's important to understand how the magnification factor is coming about. You essentially multiply the magnification factors, or the ability of the lenses to enlarge the image. Now, in most cases, the eyepiece, the ocular lens, is going to be a 10x objective, and the objective lens is essentially going to be a variable lens that you turn, and you can choose a 4x, a 10x, or a 40x. So you would multiply a 4x objective times the 10x ocular eyepiece lens, and you get a magnification of 40. A 10x objective times the 10x eyepiece would give you 100. A 40 times objective times the 10x objective would give you 400. Now, if we're using this with our eye, looking through the microscope, we would use this. But when we look at these images, it's the same basic idea, because instead of using our eye, we're using a camera, which is going to be collecting and creating digital images, but using the same basic concepts of magnification. So we can make the image larger. Okay? But the second component we're talking about is resolution. We want to have a clarity and richness of detail within the image. We don't want to have a large fuzzy image where we can't pick up and understand what's going on within it. And so the concept of resolution, in essence, is a complicated way of saying it's the minimum distance between two separate objects where the objects can be recognized as separate and distinct from one another, as opposed to one larger fuzzy image. And so if you look at the blocks on the bottom portion of the slide, you can see that they start out as two distinct images, and they basically move closer and closer and closer together. And I'm actually cheating over there on the right-hand side and showing them overlapping, but you get an idea that if we were to enlarge this, it would be difficult to be able to determine, if we have a fuzzy image, where it is that we have two distinct objects and when they start to kind of merge together into a larger fuzzy object. Now, resolution under some circumstances can be improved. We can look at wavelengths of light, and there's a fancy formula associated with it. But basically, the concept is lambda, the wavelength of light. So if we can use shorter wavelengths of light or shorter wavelengths of illumination, we can improve resolution. And so we can use some filters to change the light properties on a light microscope in the laboratory and make things a little bit better. N is the refractive index, and so we can use things like oil immersion lenses, the higher magnification lenses that you would use in like a microbiology course to look at blood cells or look at bacterial cells. And by putting the oil there, they essentially change the properties and minimize the scattering of light and the scattering of the light path, the image path, as you're looking at it through the microscope. And you would talk about that within a physics course. And then alpha is the half angle of the cone of the light entering the lens. And it's basically making better lenses. And so what we can see is that resolution under some circumstances can be improved, but for the most part, with the light microscope, we're at the advance of where we're at, and we can improve things very dramatically. And so we're limited in the amount of resolution we can see within a microscope. Now, the importance in understanding magnification and resolution is when we start to think about the magnitude or the scale of objects that we're going to be looking at. Now, most of us are familiar with the metric scale and the macro end of it. So millimeter, relatively small, a meter, about a yardstick, drop back into the English system. But kilometers in relationship to distances. 1 times 10 to the 6th meters is the diameter of the moon. 1 times 10 to the 7th meters is roughly the diameter of the Earth. So you get an idea of the magnitude we're talking about. But we're focusing in on, in this course, is the micro scale. So looking at the metric scale, but in smaller and smaller structures, so smaller than a millimeter. So 1 times 10 to the negative third, 1/1000th of a millimeter, 1/1000th of a meter is a millimeter. That's about the diameter of an intestinal villus. Take a tenth of that, you're at the diameter of a human egg cell, human ovum. A tenth of that, you're at the diameter roughly of a red blood cell. We go 1 times 10 to the negative 6th, we're at the level of a micrometer. So that's about the diameter of a subcellular structure called the mitochondria. If we go 1 times 10 to the negative 7th, 1/10 of the micrometer, we're at the diameter of a microvillus, a structure we'll talk about later on in the course. 1 times 10 to the negative 8th, about the width of a cell membrane. And so if we want to be able to understand what's going on within the membrane, we've got to be able to come up with a resolution that will allow us to look at this. And then we get down to 1 times 10 to the negative 9th is a nanometer, 1 times 10 to the negative 10th is an angstrom, and this is basically the width of a hydrogen atom. And so you get an idea of the scale that we're looking at and recognize that we're probably not going to get down to the level of being able to look at atoms, but we can understand how these things are going to be related to one another. Now, in comparison of resolution, the human eye, under the best circumstances, can get down and discriminate objects that are maybe a millimeter, maybe a tenth of a millimeter apart. And so this corresponds to an intestinal villus down to the level of about a human egg cell, a human ovum. A light microscope can get us down to about the level of 1 times 10 to the negative 7th. So we're getting down to less than the level of a micrometer, so smaller than a micrometer. So under the best circumstances, we can look and maybe be able to discriminate from a microvilli. Later on in the set of lectures, this introduction lectures, we're going to talk about an electron microscope. Electron microscope uses a different illumination source. Instead of visible light, it's using an electron beam. And because of the wavelength is much shorter, we can get a higher resolution. And so we can look at the width of a cell membrane. We can look at the cell membrane and structures within the cell membrane using an electron microscope. We don't yet have the ability to go smaller than that with a microscopy system. Now, if you have any questions, feel free to email me at hoffmanj@arcadia.edu. And again, as we mentioned previously, we're creating a mirror site for all of these resources, which is at www.tinyurl.com/AU for Arcadia University AU Histology. Thank you, and I'll see you at the next lecture.